Hello students! You began to study the fourth submodule of pathophysiology. It is called pathophysiology of blood. And today we will talk about pathology of erythrocytes and particular about anemia. On this picture you can see electronic microscopy of blood cells. These are erythrocyte, platelet, and leukocyte. Normal erythrocytes are non-nucleated biconcave discs. They are round, have a small area of central pallor. The erythrocyte's cytoplasma contains hemoglobin. As the relative amount of hemoglobin in the red cell decrease or increase, the area of central pallor will also decrease or increase. The main function of erythrocytes is oxygen transport. The erythrocytes are formed in the bone marrow. In their maturation in the bone marrow, erythrocytes normally go through several cellular forms, from the stem cell, myeloid stem cell, normoblast, reticulocyte, and erythrocyte. Such type of erythropoiesis is normal and called normoblastic or erythroblastic. But in some cases, for example, in B12 deficiency anemia, we can find disturbance in maturation of erythrocytes. Abnormally large erythrocytes, such as megaloblasts with nucleus inside, can be found in peripheral blood. Such type of erythropoiesis called megaloblastic. So, red cell development involves the proliferation and differentiation of committed bone marrow cells through erythroblast or normoblast stages to reticulocytes, which are released into peripheral blood and finally become erythrocytes. Erythropoiesis, or the process of red blood cell formation, is dependent on the release of erythropoietin from kidneys. The breakdown of hemoglobin takes place in spleen. The hem molecule, which is released from erythrocytes, is converted inside macrophages into indirect bilirubin. Then this indirect bilirubin released from macrophages into peripheral blood and transported to the liver. The next question is about pathological forms of erythrocytes. Abnormal forms of erythrocytes, which are found out in peripheral blood smear, can be divided into regenerative forms and degenerative forms. Regenerative forms, or the cells of physiological regeneration. This is unmaturated forms which are found out in blood at different types of anemias. They include reticulocytes, polychromatophilic cells, and normoblasts. In normal state, we can find reticulocytes in peripheral blood, but not more than 2%. Degenerative forms. The first disorder is anisocytosis. This is variation in cell size. In peripheral blood, we can find normocytes, microcytes, macrocytes, and megalocytes. The measure of the size of individual red cell is represented by MCV index. It means mean cell volume. The normal rate of MCV is from 80 to 100 femtoliter. And here on this picture you can see different size of erythrocytes. So this is normocyte, microcyte, small erythrocyte, and macrocyte, large erythrocyte. Next disorder is poikilocytosis. It is variation in cell shape. And on this picture and on the next picture you can see different shapes of erythrocyte. So erythrocytes can be not only like big concave discs or round, but they can have another shape like target cells, spherocytes, ovalocytes, sickle cells, acanthocytes, and others. Next form of degenerative erythrocytes is anisochromia. This is variation in color of RBCs. The color of RBCs depends on maintenance of hemoglobin in erythrocytes. The color of RBCs, or we call it chromicity, can be measured by color index. The normal rate of color index is from 0.85 to 1.05. And here you can see hypochromic erythrocyte. They contain less hemoglobin, and the zone of pallor is large. Hyperchromic erythrocytes, they contain more hemoglobin and they don't have the zone of central pallor. Next disorder is the presence of pathological inclusions in erythrocytes. So we can find jolly bodies and Cabot rings. Jolly bodies are spherical blue-black inclusions of red blood cells. They are nuclear fragments of condensed DNA. And Cabot rings, here you can see Cabot, erythrocyte with Cabot ring. So these are the rests of nuclear membrane. Such pathological inclusions we can find in B12 deficiency anemia. And the last disorder is the presence of cells of pathological regeneration. The presence of these cells represent the stages of a red blood cell before it matures. They are not normally found in peripheral blood. So, megaloblasts and megalocytes they can be found only in B12 deficiency anemia. Megaloblasts contain nucleus, and megalocytes, these are large erythrocytes without nucleus. The MCV index of such megalocytes will be more than 120 femtoliter. And now we will talk about anemia. Anemia is the most common disorder affecting erythrocytes. Anemia, this is the decrease of erythrocytes amount and hemoglobin amount in unit of blood volume, which is accompanied also by qualitative changes of erythrocytes. So we can say that anemia, this is the quantitative and qualitative changes of erythrocytes. And here you can see the picture of normal amount of red blood cells in peripheral blood and anemic amount of red blood cells. Anemias can be classified according to the next classification. The first classification is pathogenic. According to this classification, all anemias are divided into post-hemorrhagic, hemolytic, and anemia as a result of erythropoiesis disorder. Next classification is according to the types of anemia, according to inability of the bone marrow for red cell regeneration. So, according to the ability of bone marrow to red cell regeneration, anemias are divided into regenerative, when the amount of reticulocytes is from 2 to 10%, hyperregenerative, when reticulocytes are more than 10%, hyporegenerative, when the amount of reticulocytes is from 0.2 to 2%, and aplastic anemia or aregenerative, when the amount of reticulocytes is less than 0.2% in peripheral blood. Next classification is according to color index. Here you can see the formula of color index and the example how you can count it. All anemias can be divided into normochromal with normal color index, hypochromic when the color index is less than 0.85, and hyperchromic when the color index is more than 1.05. According to type of hemopoiesis, anemias are divided into normoblastic and megaloblastic. According to the size of erythrocytes, anemias can be normocytic, microcytic, macrocytic, and megalocytic. First type of anemia, which we will discuss, is post-hemorrhagic anemia. It can be acute and chronic. Acute post-hemorrhagic anemia. The main causes are next: wounding of vessels, gastrointestinal bleeding, dysmenorrhea, rupture of tube uterine, and lung tuberculosis. This type of anemia develops in 3 stages. First is acute bleeding phase. In this phase, there is an equivalent loss of erythrocytes, hemoglobin, and plasma. Second phase is the phase of hemodilution. With acute blood loss, water shifts from the interstitial to the intravascular compartment, causing hemodilution. The third stage is regenerative phase. Reduction in the oxygenation of kidneys triggers the production of erythropoietin with the following stimulation of erythropoiesis. The count of reticulocytes can increase up to 20–30%. This anemia at the third stages can be characterized as hypochromic and regenerative. Chronic post-hemorrhagic anemia. The main causes are stomach ulcer, cancer of stomach, hemorrhoid, uterine bleeding, renal bleeding, and nasal bleeding. Chronic blood loss leads to the loss of iron. That's why this type of anemia have the characteristics of iron deficiency anemia. And we can say that this anemia is hypochromic and microcytic and hyporegenerative. Next type of anemias according to pathophysiological classification, are anemias as a result of erythropoiesis disorder. They can be divided into iron deficiency anemia, B12 deficiency anemia, hypoplastic anemia, and sideroblastic anemia. Hypo or aplastic anemia. Aplastic anemia is the result of failure of production, suppression, or destruction of stem cells in the bone marrow, which leads to the decreased generation of erythrocytes, leukocytes, and platelets. So we can find that in this type of anemia, not only erythrocytes are decreased, but also leukocytes and platelets. And this state we call pancytopenia. Also, we can find the absence of reticulocytes. In this case, it will be aplastic anemia. Hypo or aplastic anemia can be acquired and can be inherited. Acquired forms can be caused by physical factors like ionizing radiation, chemical agents, often by drugs, and biological factors, for example, by hepatitis B virus. But in most cases, acquired hypoaplastic anemia can be idiopathic. That means that the cause of this anemia is unknown. Inherited form of hypoaplastic anemia is called Fanconi anemia. This is a disease with an autosomal recessive mode of inheritance. B12 deficiency anemia. The synonym is megaloblastic anemia. This anemia is characterized by an abnormality of hematopoiesis known as megaloblastic type of hematopoiesis. The commonest cause of megaloblastic anemia is deficiency of either vitamin B12 or folic acid. These substances, which act as coenzymes, are important for normal cell division. That's why in this type of anemia, cells cannot divide in bone marrow, and it leads to the appearance of big hyperchromic megalocytes and megaloblasts in peripheral blood. The main causes of vitamin B12 deficiency anemia. These are malabsorption of B12 due to the deficiency of internal Kessel factor. The place of production of Kessel factor is stomach. Then, disturbance of absorption of vitamin B12 in intestine. It can be in case of ileal resection and infestation with worms. Lack of vitamin B12 in food, but it can be only in rare cases, only in strict vegans. And the fourth cause is increased requirements of vitamin B12 or folic acid in pregnancy. This is the blood smear of B12 deficiency anemia. We can find hyperchromic erythrocytes without the zone of pallor in the center and megaloblasts. These are erythrocytes, big erythrocytes with nucleus. Also, in this type of anemia, we can find the cells with pathological inclusions in peripheral blood. These cells are Jolly bodies and Cabot rings. These cells can be found only in B12 deficiency anemia. The next type of anemia is iron deficiency anemia. It is the most common anemia affecting persons of all ages. About 80% of all anemias are iron deficiency ones. Iron deficiency anemia may be caused by chronic low-grade blood loss, such as heavy menstrual bleeding or bleeding from the gastrointestinal tract. Low dietary intake that is important in infants before weaning, especially in developing countries. And disturbances in iron absorption. The characteristic of this type of anemia is the next. Anemia is hypochromic because of the lack of hemoglobin, microcytic, hyporegenerative. And this is the blood smear of iron deficiency anemia. We can find microcytes and anulocytes. These are erythrocytes that are colored only on periphery. Hemolytic anemias. Hemolytic anemias develops as a result of erythrocyte destruction prevailing over erythropoiesis. The destruction of erythrocytes we call erythrodiuresis or hemolysis. All hemolytic anemias are characterized by an increased rate of RBC destruction and compensatory increase of erythropoiesis that results in reticulocytosis. The main clinical feature of hemolytic anemias is jaundice. After the erythrocytes destruction, hemoglobin released from erythrocytes. The most of hemoglobin which is released into the blood from destructed erythrocytes joins haptoglobin in the blood and transforms into indirect bilirubin in spleen macrophages. The third part of free hemoglobin in the blood passes through the renal filter and appears in the urine, cause hemoglobinuria and darkness of urine. And on this picture you can see how hemoglobin released from the destructed erythrocytes. Inside macrophages it is transformed into indirect bilirubin. Then this indirect bilirubin in blood connected with albumin, and this complex albumin-bilirubin indirect is transported to the liver. As bilirubin is the pigment, the increasing of it leads to the yellowing of skin and mucus. All hemolytic anemias are divided into acquired and inherited. Acquired hemolytic anemias can be caused by mechanical damage of erythrocytes, immune hemolysis of erythrocytes, toxic hemolysis, and infectious hemolysis of RBCs. Inherited hemolytic anemias are divided into three groups. Inherited membranopathia, this is the disorder of red cells membrane. Inherited enzymopathia, this is the defect of enzymes inside erythrocyte cytoplasm. And inherited hemoglobinopathia. This is the inherited disorder in which an abnormal hemoglobin is produced. Enzymopathies. The most common cause is the deficiency of glucose 6-phosphate dehydrogenase. The deficiency of this enzyme reduces the ability of red cells to protect themselves against oxidative injuries and lead to hemolytic disorders. Some drugs can initiate the development of enzymopathia. These are sulfonylamides, antimalaric medicines, aminosalicylic acid, vitamin C and K, aspirin in large doses. Membranopathia. This disorder is also known as hereditary microspherocytosis or Minkowski-Shaffer disease. Membranopathy is a genetic defect of erythrocyte membrane proteins. The main one is spectrin. Due to its contractile properties, the erythrocyte membrane keeps the form of biconcave disc. If the content of spectrin decreases or its polymerization occurs, the erythrocyte cytoskeleton is destroyed. It leads to membrane tension decrease and provokes transformation of normal biconcave discs into spherocytes. Membrane changes predispose erythrocytes to hemolysis by macrophages. And this is the blood smear of hemolytic anemia, inherited membranopathia. We can find many spherocytes, microspherocytes in peripheral blood and reticulocytosis. This is also one of the types of inherited membranopathy. This is hereditary elliptocytosis. And here we can find elliptocytes. Hemoglobinopathy. Hemoglobinopathy is connected with the synthesis of hemoglobin, which is abnormal or not typical for a given age. Hemoglobinopathy can be qualitative or quantitative. Qualitative disorders of the primary hemoglobin structure. This is caused by the replacement of amino acids in hemoglobin molecule. This disorder leads to the development of sickle cell disease. Quantitative disorders of the secondary hemoglobin structure leads to the replacement of alpha or beta chains of hemoglobin, and then thalassemia develops. In sickle cell disease or qualitative disorder of hemoglobin, hemoglobin S is formed because of glutamic acid in the third chain of hemoglobin is substituted by valine. And that's why sickle cell erythrocytes are formed in blood. And here is the blood smear where we can find sickle cells. Thalassemias. The thalassemias are a heterogeneous group of inherited disorders caused by mutations that decrease the rate of alpha or beta globin chains. The beta thalassemia is called Cooley anemia or Mediterranean anemia, are most common in Mediterranean populations of Italy and Greece. Alpha thalassemia are more common among Asians. Alpha and beta thalassemia both are common in Africans and African Americans. In thalassemia, we also can find some special erythrocytes. They are called target cells. And here are the target cells in blood smear. That's all for this topic. Thank you for attention.